So I wanted to thank everybody. My name is Jonathan Waller, and I work at the Operations Center for the Children's Brain Tumor Tissue Consortium at the Children's Hospital of Philadelphia. I wanted to thank everybody for being here today. This is the CBTTC's first Facebook Live panel discussion. We're holding this right at the tail end of Brain Tumor Awareness Month in May. As we've seen from all the organizations that have been represented here today, some amazing resources and information have been shared this month. I just kind of wanted to give some context for why we're talking today. Brain and CNS tumors are the most prevalent form of pediatric cancer in children under the age of 19, and they're currently the leading cause of cancer-related death among children and adolescents in the US and a number of other countries around the world. Brain tumors affect coordination, movement, personality, cognition, and a number of other critical brain functions for all people. However, on the growing brains of children and adolescents, their effects are more significant and longer lasting than those in adults. Despite all this, only 4% of federal funding for cancer research is devoted to the study of all forms of childhood cancer. And in the past 30 years, only 4 FDA-approved drugs and 1 medical device have been developed to treat all brain tumors. And there's never been a drug developed and approved specifically for malignant brain tumors in children. So in this context, today's topics are covered by a panel that's really encouraging. We're joined by a group of scientists, clinicians, and research supporters who've dedicated collective years of time and effort to advancing brain tumor research in children, and are starting us all off on some really promising efforts to accelerate to accelerate the development of new clinical trials that hope to find new therapies to tackle a variety of childhood brain tumors. We are live on Facebook, so those watching via Facebook Live can leave comments or questions in the comment section, and I'll do my best to pose these questions to our panelists. So I thought I'd start us off with a quick round robin discussion with each of the folks here. I'm just gonna go through alphabetical order style, and if our panelists can just give name, title, and the organization that they're representing today. So we'll start with Dr. Jess Foster.
Hi, so as you said, I'm Dr. Jess Foster. I'm a practicing neuro-oncologist at the Children's Hospital of Philadelphia, but I'm also a physician scientist and spend about 80% of my time in the lab doing translational research.
Thanks so much. Next, we have Kristen Gillette.
Hi, I'm Kristen Gillette. I'm the founder of the Kortney Rose Foundation. Our daughter Kortney passed in 2006 from a diffuse intrinsic pontine glioma. And since then, we've been supporting all the work of these fine people and the growth of this Children's Brain Tumor Tissue Consortium.
Thank you so much. Next, we have Al Gustafson. Oh, Al, you're on mute there.
Sorry about that. Al Gustafson, I'm from the Swifty Foundation. I'm in Chicago. We lost our son, Michael, in 2013 to medulloblastoma. And since that time, I've been involved in supporting pediatric brain cancer research, very invested in CBTTC, and our other main focus is postmortem collection.
Next, we have Amanda Haddock of Dragon Masters.
I'm Amanda, and my son David passed away from a glioblastoma in 2012. He was very interested in research, which led us down a path of supporting research, but more specifically, we're interested in that big data space and how we can accumulate enough information for researchers to really start learning things about the disease that they have been unable to discover in the past.
And next we have Dr. Eugene Hwang.
Hi, my name is Dr. Eugene Hwang. I'm a pediatric neuro-oncologist at Children's National Hospital in Washington, D.C. I'm the Associate Chief of Oncology there. And really, besides trying to help every patient that I see, I'm also really interested in helping to run and write some of those clinical trials that we hope will move the needle for all patients with pediatric brain cancer.
Great. Next, we have Dr. Seyed Nabavizadeh.
Hi, everyone. I'm Ali Nabavizadeh. I'm a pediatric and adult neuroradiologist and also a nuclear radiologist. And what brings me here is my passion for integrating imaging into CBTTC and into all these wonderful precision clinical trials. So that's why I'm here.
Thank you so much. Joining us all the way from Zurich, Switzerland right now, we have Dr. Javad Nazarian. Javad, I think you may be on mute. If you can hear us. Oh, I think we may come back to you, Dr. Nazarian. And then we'll just highlight Dr. Mike Prados is here.
Yeah, hi, my name is Mike Prados. I'm a neuro-oncologist with a long history of focusing on both adult and pediatric neuro-oncology, and am from the University of California, San Francisco. I co-lead the Pacific Pediatric Neuro-Oncology Consortium, which is an early phase clinical trials consortium for children with brain tumors, and co-scientific chair for this Children's Brain Tumor Tissue Consortium.
Thanks so much for being here. And next we have Dr. Adam Resnick.
Hi, this is Adam. I direct the Center for Data-Driven Discovery in Biomedicine here at CHOP, and have the privilege to be scientific chair of PNOC and co-chair with Mike of the CBTTC.
Okay. And finally, we have Dr. Nick Vitanza.
Yeah, hi, I'm Nick Vitanza. I'm a pediatric neuro-oncologist at Seattle Children's Hospital and the Fred Hutchinson Cancer Research Center in Seattle. I take care of new patients with DIPG and other malignant brain tumors. And also I'm the CNS CAR T-cell lead at Seattle Children's and run 3 open CAR T-cell trials specifically for children with brain and spinal cord tumors, and then do DIPG lab work within Jim Olson's lab at the Fred Hutch.
Great, so obviously, as folks can see, we've got an unbelievable panel of guests today. Thank you all so much for being here. So I thought I'd kick us off with asking Dr. Prados and Dr. Resnick a little bit about some context. If you could work together to tell us a little bit about, the CBTTC and PNOC, especially how their missions overlap and how they first began doing this sort of cross-collaboration work.
Maybe I'll start, Mike, and then hand it to you. So for many of us who have been trying to drive the discovery process for pediatric brain tumors early on, and still, the challenge ends up being the need to really coordinate the cross-institutional efforts on behalf of gaining knowledge so that we can then translate into clinical care. And so really the process of building the required infrastructure for collecting specimens in standardized fashion to generate large data sets like Amanda had highlighted, making them rapidly accessible on behalf of all the bench science and biology that needs to be done, was the first mission for the CBTTC. But all of those efforts were never data for data's sake. They were all geared towards clinical translation. And just like we need to coordinate efforts across institutions on behalf of biospecimen and data generation, equally, no single institution can run a clinical trial or advance clinical care on its own. And I think this is where there's the opportunity to really connect with organizations like PNOC. To you, Mike.
Yeah, so it's interesting. PNOC actually was formed out of, and I've said this many times before, out of frustration of the pace of clinical research that was being supported by the NCI. Not to put the NCI down, but it was really over 20 or 30 years, for me, a much too conservative approach, particularly in pediatric brain cancer, and in particular for DIPG, certainly, but for other malignant brain tumors in children. So we formed PNOC to, I think, enrich the landscape for doing high risk, high reward type of clinical trials, early phase trials that were tissue-based, precision-based, personalized approaches so that we could assess whether the therapies and the strategies and the hypotheses that we had actually made sense, and would also, whenever possible, deal with futility so that we're not doing a one-size-fits-all trial for 5, 6, 7, 10 years, never knowing whether what we're doing works or not. And so we gathered like-minded individuals, many of whom are here, who felt the same way and developed this enterprise called PNOC. Part of the issue for PNOC, of course, is the large data that is necessary to inform clinical decisions and prioritizations. I mean, anything that comes from the lab is exciting, but does it really reach the threshold where we want to bring it into a child who has such a high unmet need? So we wanted to engage in an open source way, in a real-time way, all of the data, the information, and the knowledge base that was out there. And CBTTC is obviously the place to be for that, because their mission is basically to share. And ours is the same for on the clinical side of research. And part of the other aspect I think that's really important is to kind of strategically think forwardly about what we're doing rather than ad hoc. And I know that much of what we do in the past or have done in the past is because of either serendipity or the fact that something is there and we want to use it, as opposed to actually thoughtfully thinking about the disease, the landscape of that disease from a biology standpoint and over the course of a child's life. And that's all of the things that we really care about in terms of the engagement with the CBTTC and PNOC coming together to form that philosophy.
Great. Thank you so much, doctors, for that just bit of context there. And I want to just kind of jump straight in. You know, the CBTTC and PNOC have worked together to form these clinical trial working groups that are really combining the collective talent of clinicians, researchers, and research supporters, foundations. And so I wanted to kind of talk first about our DMG, our diffuse midline glioma working group. We've got a number of folks who are representing that group here today. Dr. Vitanza, we have Dr. Foster, Dr. Nazarian, but I also wanted to actually reach out and highlight Kristen Gillette, who, you know, we have a unique model where foundations and research supporters are joining as disease champions. And so, you know, Kristen Gillette and the Kortney Rose Foundation, as she has said, have been supporters of the CBTTC's mission basically since the very start. And so, you know, curious with Kristen, you know, what were your hopes in collaborating with this working group as a disease champion? And why do you think it's so important for patients, families, and foundations, and just everyday people to be aware of this research into childhood brain tumors? And you are on mute there, so if you just want to unmute yourself.
That would help. We have been fortunate in the sense that in 2006, when Kortney died, nobody even knew what if you said the word or the acronym DIPG, which is diffuse intrinsic pontine glioma, which is a tumor in the brainstem and is fatal in every case, nobody knew what you were talking about. I wanted to, when I formed my foundation, I wanted to support research for DIPG, but I couldn't find any. Nobody was doing it. And here it is, the #1 killer, you know, by brain tumors, and nobody could get funding to do any research. So we fund research for all brain tumors. But then a few years later, I met Adam Resnick, and he said, I hear you have an interest in DIPG tumors. And that was the beginning of now really seeing in 10 years the difference and the amount of work that these researchers are finally doing on DIPG and other midline gliomas that are really important, that haven't ever been looked at before. And it's because of foundations like ours and many others that are funding this research. If it weren't for us, this research would not happen. So anybody out there who has a small foundation or just wants to do a fundraiser for DIPG research, it's available to you. And I encourage you to get involved with the Children's Brain Tumor Tissue Consortium and PNOC, because all these people are sharing, they're doing it right. And, you know, we will only support research where people are sharing. And we're so fortunate to have been involved with this effort since it started. And it really gives back to a parent who's lost a child the little bit of control they might have in helping other children in the future.
Maybe Javad can chime in, Jonathan, because I think, when Kristen approached me back then, the ultimate vision of what I'd hoped could happen of really bringing together, right, institutions and scientists on behalf of a common coordinated effort, I think was really something Javad and Sabine Mueller had really championed. Maybe Javad, you can talk a little bit about how
Sure.
it work.
Right. Adam, you can hear me now, right?
Yes, you're coming in loud and clear. Thank you so much.
Good. Thank you very much, Jonathan. This I've witnessed over the past 10 years really a beautiful evolution of so many forces coming together to do what we should have been doing for a long time for pediatric brain tumors, especially as Kristen said, for the deadly forms of pediatric brain tumors, DIPGs, in this particular case. 10 years ago, if I wanted to do any type of research, we didn't have specimens to work with. Then came CBTN or CBTC, where you have access to a fantastic biorepository. It's not only frozen specimen, but also well annotated genomic data, molecular data that we can use. But then the question was 5 or 6 years ago for me as a scientist who was the recipient of these fantastic donations that children had made to decide to donate this post-mortem to us. Then the question was that, what is the clinical link between the samples that I have? When can I make that difference by using the specimens that I have? And we've been very lucky at Children's National, working with Gene and working with other colleagues to be a part of the PNOC, where the goal is to take the data from the CBTC to translate into PNOC, where there's a truly existing clinical trial that needs real-time data to go into the clinical trial. And as of recently, as you mentioned, the DMG Act has been a new form of a live and real-time collaboration that has been made as a consequence of PNOC and CBTCC, CBTN working together. The idea was that, could we have a group of like-minded researchers across the globe that could be working together and sharing data real-time in the context of bi-weekly calls, and focus on one thing: what are the best combination drugs that I can take into clinic for a particular indication, in our case, DIPG. And of course, with any type of research that you start, you don't have enough resources, funding specifically, to initiate that study. NIH, NCI are not well suited for new ideas that doesn't have enough preliminary data. That's where foundation, including Kortney Rose Foundation, has been fantastic. I've been very, very lucky to be the recipient of many of the hard work that I've been doing. The idea was that if the DMG Act, which is a group of scientists and clinicians, has ideas, they work together internationally without the need of publication. If there's a financial need associated with this effort, foundation are linked into it, which was a good example of it was the funding that we received through the Kortney Rose to push some of these ideas forward. The DMG Act now consists of 18 different institutions from Australia to Europe and all across the United States. And again, we have a single mission in mind. How can we take all of the resources that CBTC and PNOC has provided to take data to the next level, to take it to the clinical use?
Sorry about that. I muted myself just for a moment. That's really a beautiful summary. Thank you so much, Dr. Nazarian. And I really kind of wanted to also make sure that I pitch this to Dr. Foster and Dr. Vatanza. You're both relatively younger researchers and sort of joining and making really fresh, great new discoveries within your fields. And I wanted to hear from you both. There's a lot of really great, exciting work that's going to be planned and done through these clinical trial working groups. And I'm wondering to see if either of you want to comment on some projects that you're excited to be taking on, or the potential that you see in these working groups to kind of do work or make progress that has yet to be made in these really challenging diseases, diffuse midline gliomas?
Jess, do you want to go first?
Sure. Yeah, I think I have been really, really excited to be a part of CBTTC and PNOC in our group, and the DMG Act as well, and to get to know Nick better. Nick and I do pretty similar work. Actually, we both are focused on CAR T cells for pediatric brain tumors, with a big focus on DIPG, amongst others. Pediatric brain tumors. And so it's been really, really wonderful to have the resources together to come together. And he and I are both part of one of the upcoming big grants that we're putting together. So we've been able to collaborate across institutions to really help to try to put together the best CAR T cells. And I don't know if folks on Facebook that are watching are familiar with CAR T cells, but this is a form of immunotherapy where you basically take a patient's own immune cells from the body and you reprogram them so that way they can go out and seek cancer cells and attack them. And so Nick and I are both working on that, but at totally different institutions. We have different targets we're going after, but we're putting our minds together and trying to figure out what's the best way to both go after the targets, study them, figure out the best ways to monitor and make sure that we're doing the best we can as far as treating these patients. And Nick, I'll open it up to you to add anything else.
Yeah, I'll take the being young as a compliment, I guess. I remember back when I was in high school in the year 2000. No, I'm just kidding. I echo a lot of what Jess said. It's true. It's really wonderful that we're able to work together. I think we are very lucky to have Adam, Javed, and Mike's leadership, and they've done a lot of work to break down silos that were between institutions and that prevented things from moving more quickly, and have been very, very generous in bringing up junior investigators, supporting multi-institutional collaborations. And there's certainly an older way of thinking where you want all the grants to come to your hospital, you want all of the funding to come to your hospital, and there is a advantage for your research career to actually box out other groups. And I think that we have all said that that is unfair to patients, it's unfair to the families that are interested in us doing work, and how can we make connections to prevent that from happening. So Javed started a weekly research meeting for DMG, where literally every week we present preclinical laboratory data before it's published, before we show it at a conference, can critique each other's work, know of each other's work in a way that we don't have to be afraid that other people will steal those research projects or write grants, and that we can work as a community to inform each other as quickly as possible, thinking that progress is the most important thing. And from a CAR T specific perspective, I think that I'm lucky to get to work with Jess, and that we're both in a unique position where we both see patients with DIPG, both do laboratory work, and are both involved with clinical trial design. And so that we can bring the things we see from a patient and the clinical things we know will be necessary, incorporate them into a clinical trial, and have what seems like would be overwhelming and not possible actually makes for this really efficient, streamlined approach where we can go bedside, bench, clinical trial, and back, and incorporate information in real time.
Amazing. It's so exciting. I want to make sure that I'm able to represent as many of the working groups and folks possible. So I am going to kick it back to Mike Prados, but also Al Gustafson. Al, you are disease champion for our medulloblastoma group, and Dr. Prados, you sort of have a hand in practically all the working groups that we have today. So I kind of wanted to kick it over to you again. Al, I think it's really unbelievable that you and Kristen and Amanda, Haddock, and a number of other foundations are signing on to be disease champions. I wanted to see if maybe you could kind of comment on, what does it mean to you to be a part of these working groups, and what are your goals in specifically informing these scientists on their work? What is it that you want to get across, from a patient side that is reflected in this work, and just sort of how do you hope to influence?
Okay, I think I'll take it. I heard most of your question, Jonathan. Thank you. in terms of informing the scientists, since my last science class was high school biology, I'm not going to be able to be too informative in terms of the science, but I think what I've discovered in my few months participating in the Medulla group, that as a parent of a child who had medulloblastoma, I do bring a unique perspective and an experiential knowledge that can be very helpful in informing the work of any proposed trial. I think what attracted me to the participate in the working group, first and foremost, was the opportunity really to have access to some of the conversations and try to understand how the science evolves. As a funder, like most of us that are doing this, we came to it through tragedy and trying to make some good grief out of our experience. And again, not really understanding the product that we're investing in, at least for me, that's true. And I've been at this for 6 years now. So to have the opportunity to be involved in some of these conversations is extremely helpful. I feel like I've got my handprints a little bit on what's happening in the conversations in the working group as they ask for parents' perspectives. I get to know the researchers a little better. I understand the evolution of the work a little more. And then ultimately, when it comes time to make an investment, I feel more informed and I trust the people that I'm investing in. And I think in addition to that, it invites me really to make a difference, I think, where I'm able to, and that then becomes that allows me to become an ambassador, if you will, for the research that's taking place. We currently are working on a TNF trial in the medulloblastoma group that, none of us funders really have the capacity to fund these trials individually, given our means. So we have to do it together, much like the science is done in through CBTTC and PNOC. So we really, I think it invites philanthropy to think around the same model. And so, again, where I think we can make a difference then is that we can take the knowledge that we've gained in these working groups and then go out and invite other potential funders to participate and build collaboration around the funding structures that support the work, which, again, the private dollars are in limited supply. And the more we can do to corral those dollars in a focused effort, the more effective they're going to be.
That's such a great summation.
Can I make a comment? I mean, I think, how you've been embedded with that group has been amazing and it's really enriched it. The other part, though, that I think that you didn't mention, and I think it's worth saying, is that we want to bridge, we want to make a difference, right? And one of the things about this trial, the TNF trial, is doing a research biopsy, which is really challenging from a regulatory standpoint. And IRBs are probably going to be uncomfortable with that. So when patients and families come together and say that, like you said, you trust in the research enough to support something as paradigm shifting in some ways as a research biopsy in a child, which is going to take some effort and explaining to IRBs, your voice and all the voices of the advocates are critical for that. And as part of this working group, actually, we're going to have a discussion, I think next week or so, where families are going to talk about the issue of research biopsies and galvanize kind of a consensus and a narrative and a discussion that we can bring forth, I think, to make sure that we make that happen, because the trial requires that. I mean, in order for us to really know what we're doing in real time. So I think that that's really a critical part of what you also bring, besides the funding aspect, but that perception of what we're doing and the relevance of it. I just thought I'd put that out there.
Thank you. And actually, before we move on to some of the other panelists, I do want to actually ask a question for you, Dr. Prados and Adam, just very briefly. We've got a captive audience here. I'm looking at the Facebook comments. We've got folks from Phoenixville, Pennsylvania, Washington, D.C., Wichita, Kansas, Chicago, Florida, really across the U.S. And a lot of the folks that follow us on Facebook are, not researchers, but foundations and families. And, the hope is that the message that they're receiving here is that really anybody can be a part of this work. And, if you could talk to the folks who are watching today and let them know, how they could be a part of not only the working groups, but really just the work of these two foundations, or these two rather scientific consortia, what would you like to say to them, and how would you encourage them to be a part of this?
Well, I mean, I'll just briefly say something, but and then Adam can add to it. So PNOC, which is an early phase clinical trials consortium, is almost entirely funded by philanthropy, almost entirely. To do clinical trials requires an investment by the public as a priority, right? So do we really care enough to do what we're doing? Because the NCI, as you mentioned, is not supporting. And the years of life lost in these children is devastating, right? So we want engagement. We want as much engagement as possible. And we can provide the opportunity to engage, to communicate, and to be partners. And we need that handshake. We need that the partnerships of anybody who wants to be involved with this, particularly if you have energy, because it's an exhausting process. I know, Jess and Nick, you've got CAR T cells. That's exciting, but there's so much more to do there, right? And every aspect, every time you turn a corner, you need more resources to find the answer to the question that keep coming up to make it a better therapy for these children. So we want engagement. Join us, right? Give us a call, send an email. Let us be partners with you. Adam?
I just want to say quickly, I totally agreeing with that, and that, some of the therapies that we're trying that we don't have data yet, if they're successful or that if they'll be beneficial, but we think is so deserving of a chance. New technology and new developments are just so incredibly expensive. And our CAR T cell program at Seattle is also almost entirely philanthropic funded. And I'll use this as an example, but it would go for any one of our projects that as we bring it to multiple institutions, we need more support to be able to open it at other places. And Mike and Adam and others have done such a great job of putting an infrastructure in place that allows these things to be available across the country. So there's also less of a disruption to children's lives. And so by opening things at multiple institutions, the extra support allows us to do that.
Yeah, thanks, Nick. And I completely agree with what both you and Mike, just iterated. And one of the things that I'm sure as an audience you're hearing is that collaboration and engagement and partnership are thematic core beliefs. And, even as we are thinking about brain tumors, the reality is that brain tumors won't be able, brain tumor research communities themselves will not be able to solve cancer on their own. So even work in CAR T leverages work in other disease contexts. So for us, as a community, what I would say is, there is no boundary. Even in the context of thinking about pediatric brain tumors, there aren't enough of us to do all the research that's required. So we need cross-disease analyses, cross-modality analysis, cross-technology opportunities to intersect. And as Mike highlighted, right, the door is wide open for that. So the boundary even goes beyond brain tumors to look for other pediatric cancers in adults, and looking for ways to leverage any aspect of the discovery process that we can bring to bear on behalf of translation for children with brain tumors.
Thank you so much, Adam. Actually, I'm gonna keep on Adam, but also bring in Amanda Haddock. Amanda is a disease champion for our craniopharyngioma working group. Adam is also a member of that working group. I'm wondering if one or both of you could sort of explain, what sets craniopharyngioma apart from some other forms of childhood brain tumors, and what are you hoping to contribute to this working group? What is in an ideal world your role and the role of folks of your ilk, whether they be foundations and everyday folks, patient families, or on Adam's side, researchers across different institutions. How can how can folks inform on the work that you're doing?
Maybe I'll start, Amanda, and then hand it over to you. The craniopharyngioma working group actually leverages, opportunities that, I'll have Amanda further iterate on, which is that, as everybody has highlighted, almost the entirety of most of our efforts begin with philanthropic initiatives. But we've also made inroads into partnerships with the NIH and NCI. And it's through that effort that we began a pilot initiatives in looking at proteomic data, essentially, what proteins are expressed in different tumors, and began identifying potential targets in this way in craniopharyngiomas. Craniopharyngiomas are a type of tumor that, oftentimes is deemed curable because you can resect the tumor and treat the patients, but it is in a part of the brain that then leaves very long-lasting effects on the child. So the key is really how to prevent, the damage to the brain that currently is imparted through surgery, through more targeted approaches. But one of the really exciting things about the craniopharyngioma working group is that it really has broken down barriers between philanthropically funded efforts and how they can partner with NCI initiatives. Namely, here is CBTAC, a proteogenomic initiative within the NCI. And then I think what I'd love Amanda to talk about is, given your role and activities that you really championed on the advocacy side, intersecting and interacting with the NIH, what are your thoughts about how we can even move that forward as we tackle craniopharyngioma or any other tumor?
So I'm going to back up just a little bit because I think that how I came to be on the craniopharyngioma group is a little interesting. So my son had GBM, and we've already talked about high-grade gliomas. And in the process of just talking with these different researchers and different families, it became really apparent to me that, in a sense, we were fortunate that with GBM or with DIPG, there were these well-established communities by the time my son was diagnosed. There were well-established communities of someone, right? There were at least a couple of researchers out there working on something. There were at least a couple of other families that we could find that had some similar experiences. But the thing that struck me around these other more rare tumor types, and maybe not more rare, but less organized groups around them, both of those factors, was that there really is strength in numbers. And it's not just about the type of tumor that my son had or the type of tumor that kids in my hometown have, but that we can all empower research for all of these types of tumors. And indeed, we're starting to find, connections between these different tumor types, right? There's mutations that affect these different types of brain tumors. And so by studying those in silos, we're really missing a lot of information that could inform research across brain tumors. And to me, it was important to, even though I'm coming from a high-grade glioma world, to kind of reach out to that other community with the craniopharyngiomas and let them know that there are people working on those types of tumors. And I think those patients were, first of all, just thrilled to have somebody working on that. And they wanted to share their experiences. And a lot of times we get caught up in this narrative around how terrible it is, for a child to have a high-grade glioma. And I mean, I know that firsthand, right? My son only survived for 20 months after his diagnosis. But it's a different kind of terrible for these kids that are, they are living much, much longer, but they have horrible weight issues and they don't have their sight a lot of times because of the effects of these tumors and just these really extremely life-limiting things that they have to deal with that, yes, they're alive, but these are still really, really serious side effects. And so I like the fact that the research that's happening within PNOC, within CBTTC, kind of doesn't leave a man behind, right? Like we can bring everyone along. We can, and for kids that maybe they're only one of 20 with that type of tumor or whatever, they're not left off in some dark corner somewhere. They're there getting the same kind, or maybe not exactly the same kind, but at least a chance at the same kind of research and the same kind of help that kids with these more common tumors have. And I'm sorry, Adam, you threw me the ball in a different direction. And I didn't go there, but
you actually took it in the perfect direction. And I just wanted to highlight that, as was mentioned earlier, there are a number of different ways that foundations and partners, community stakeholders partner with us, and working groups. Disease Champion has been one, but you have also been an amazing advocacy champion, working with the NCI and NIH. And we've mentioned them a couple of times. And, I think there is forward momentum now, especially because of some of these collaborative efforts, for engagement at the NCI. I just wanted you to comment on that as well.
Yeah. So I think that we're seeing a real shift in a lot of these types of efforts, and the NIH and the NCI are definitely going to be a big part of that, right? When we started our foundation, we sort of did it thinking we're not going to get help from the government as quickly as we want it. I think maybe we'll never get it as quickly as we want it, but lately I've seen, especially under Dr. Sharpless's lead, a real effort to involve patients and advocates from the very beginning. There's a shift in their attitude that we're not just doing things to patients anymore, right? We're doing things with patients. We're involving families from the very beginning. And I think, that's something that that everyone on this call can be really proud of, because I think CBTTC and PNOC have sort of led efforts in that direction, right? You've been partnering with families and with foundations for a long time, and really listening and really collaborating, and it's team science in a different way, right? In the past, team science has just been doctors from different departments working together, and now it really is these families helping to inform how the clinical trial is even developed, like from the ground zero up. We are involved every step of the way. And I think that it's going to make all of these projects run smoother and faster. And I do think that'll help with funding as well, right? We want to fund things we're involved with. And so we are inviting patients and families to be more involved in this so that they can help direct them and also help fund them.
Reflect on what I think everybody has been saying. I think what it all boils down for me is to understand how we're achieving our mission. It's the same for everyone on this call. And it's been the same for doctors and scientists and families for decades. That mission is to cure every child who has brain cancer. There have been a lot of really smart scientists and doctors and families who've been invested and passionate for decades. And yet for some reason, things haven't moved quickly to the point where we're able to achieve that goal for the majority of our patients, unfortunately. So why is this approach different to me? I mean, the same mantra about working together, those are not new goals. Those are not new dictums that we try to live by. The difference today that I see, and the thing that gives me hope, because we haven't cured brain cancer yet, but the thing that gives me hope is that with the efforts of CBTN and PNOC and Amanda and others who have really started to build an infrastructure that welcomes many different players to the table at the same time. It really is the fact that before there were a lot of people in a lot of boats, and some were fast and some were slow, but now this group has really created a boat that is fast and streamlined and big, and welcomes everybody in to row dynamically in the same time to really kind of beat to that finish line. I think that's incredibly important. And this involvement of the foundations and the families in that process, which has not been as typical as has been made in this particular structure, that's amazing. And for everything that Amanda and Al and Kristen and others have said, it's amazing because not only do we realize that in some ways that there have been much more efficient ways that we should have been going about our work as scientists and doctors before, but also that at the end of the day, this is really the group that we want to serve. And there's no one better enabled to inform us, to be ambassadors to the rest of the patients and the families that are worldwide at this point, connected by the internet, than patients and families themselves. And so it's this whole ball of wax put together that I think is providing an immense amount of hope, not because the science has been more evolved, although it has been, but because the way that we're approaching science and the way that we're approaching translation of that science to cures, that's what's different. And that's what, to me, is one of the most exciting things about this group.
Very well said, Dr. Hwang. Thank you so much. And actually, you were my next guest to speak to. You're a clinician at Children's National Health System in Washington, D.C., and you're working on our ependymoma working group. Amanda really encapsulated so much of why this work is so critically important. Even for children who survive and continue to grow past their brain tumor diagnosis, there can often be some really devastating long-term effects, some lifelong. I'm curious to see in your clinical practice, and to give a little bit of context to what these clinical trials may be able to do. What do you foresee could be the impact of some of these clinical trials? When we say clinical trials, we don't just mean drug therapies. We also mean new modes of surgery. We mean new modes of other non-invasive therapies, ones that are less toxic to children. And so, looking at the work of your working group, what do you hope could make a difference in the way you get to treat your patients, and potentially how some of the other clinicians on the call today could treat children patients to not only survive but thrive beyond their brain tumor diagnosis?
Yeah, thank you. I think you've asked two really big questions. One is sort of surrounding a brain tumor called ependymoma, which is another kind of brain cancer, which is very difficult sometimes for us to cure. And the other is this really difficult space that we find ourselves in, which is it's not easy to cure brain tumors. And we don't actually know exactly how hard we have to push on any given child to be able to eradicate that tumor and to cure it. And the tools that we have to date are not friendly to the normal body. So those toxicities can be temporary, but all too many of them are permanent. So maybe I'll address that question first, which is this idea of what do you do when the way that you've actually cured a child has damaged them so significantly that it impacts their quality of life going forward. I think that's really part of the Holy Grail. That's the half of the Holy Grail. The first half is to make sure the tumor never comes back. The other half is to make sure that that child is as perfect as we can possibly make them. But radiation and surgery and chemotherapy and even immunotherapies, those have a lot of toxicities to hearing and to kidney function, and maybe most importantly to the ability of the brain to learn and to think in the best way that it was designed to do. So interestingly, this group, I think, is reshifting the paradigm in many ways, but there are so many different brain tumor groups and children's oncology groups across the world and in this country that have been really working on this exact goal, which is how do we drop the hearing toxicity so that the loss that we experience with curative chemotherapy is not so much that that child needs hearing aids, or how do we lower or protect someone's ability to learn so that when they do become an adult, they're as perfect as they could have been even before the brain tumor was diagnosed. But it's a complex question, because every time you think about something to reduce the toxicity, you always have to worry about, are you giving the tumor or the cancer itself too much breathing room to really to be cured also? I think that ependymoma is one of those brain cancers, and that's the working group that I'm most involved with. But interestingly, I think that, you think about everybody getting into one boat, but there are multiple goals. There are goals of the corneopharyngioma, the diffuse midline gliomas, ependymomas, ATRTs, medulloblastomas, as Al was mentioning before. There are lots of different kinds of tumors. And one of the parts that I think is also really exciting is that PNOC and CBTN have put together groups of people, sometimes who are in multiple groups, and many of the therapies and the ideas that would inform many kinds of different brain cancer treatment. And so whilst the group that I am working most closely with is really focused on ependymoma, and Al was talking about medulloblastoma and Javad was talking about DMGs, at the same point, many of us are interrelated across those groups. And that's another part of the way that I think that this particular team has tried to integrate not only members of PNOC and CBTN, but external stakeholders, foundational spaces, and then to cross link those groups within CBTN and PNOC as well, within these disease champions and these working groups. But ependymomas are relatively unique in that whether they progress or whether they're cured, radiation is always part of the answer. and the course of that cure or that course of that relapse spans years rather than months. That's obviously a benefit for patients and families to some extent, but it also really highlights this issue of long-term toxicities where the chemotherapy or the radiation has really impacted many of these young children whose brains are supposed to be developing quickly, whose brain cells are supposed to be dividing and linking, and our therapies sort of interrupt that process. So I won't pretend to have any sort of answer, but I will say that it's a really strong focus, not only of the ependymoma working group, but of every, I think, person on this call, because it's really, that coin is really, we have to address both sides of it at the same time.
Thank you so much, Dr. Hwang.
Jonathan, if I just may add something really quickly, I think one of the most amazing aspects of these new groups that has been formed is to bring different disciplines that has been historically missing from the pile. Typically, it's been a group of physicians and scientists working together, trying to figure out what's the best medicine. But the working groups that we have now includes bioengineers who come along, and their main goal is to define the basal level of the brain network. What is a healthy brain network? And there are emerging technologies now that they can map a healthy brain and then put that in the context of a drug to assess drug toxicity. This is the work that we are doing here to assess not only the efficacy of a drug in killing tumor cells, but also how is it affecting the healthy adjacent normal cells. The artificial intelligence has come to the group now. We have groups from the NCI who have huge resources to look at different drugs and drug combinations that have come to the aid of the working groups that we have built. So is the traditional model of physicians and scientists has really changed in this new opportunity that has brought together by the PNOC and CBTN and the families who are supporting this.
Thank you so much. I want to just make a quick just housekeeping observation. We are coming up on 1:30, and we do have a number of folks who are actively working in labs in the clinic, and so to be mindful of their time, we may lose one or two of the panelists. However, we did reserve some extra time. I anticipated that this conversation could go on all day, but we've got a bit more time to go, and so any folks who need to drop off at 1:30 Eastern time, I do just want to say thank you so much for participating. But we do still have a few more questions, a few more panelists to get to. And so we welcome folks to stay on. Also, if you do need to jump off, please remember that we will be archiving this conversation on our CBTTC Facebook page, as well as our YouTube channel and our website. So thanks again. But right now, I do want to turn the panel over to Dr. Nabavizadeh at the University of Pennsylvania. Doctor, you're joining us as part of the imaging working group, medical imaging. And it's for me, it was fascinating to hear that you were joining. And I'm wondering if you can kind of explain a little bit about, how medical imaging can fit into these clinical trial development, and what exactly can be gained from a more precise understanding and effective use of these images in research.
Sure, thanks, Jonathan. And just want to mention, as you also pointed out, the imaging unit is a new unit, both to D3B, which is a core center for CBTTC and PNOC. And so one of the main missions of CBTTC has been to act as a platform for bringing together the data from many centers in order to transform pediatric brain tumor research. And it has really done a terrific job in bringing the genomic data and tissue and biospecimen data. And the fact of the matter is that all these patients have some form of imaging, mainly in the form of a brain MR, that we can bring into the research. And currently we are developing a platform that allows us to capture all these images and curate them and organize them in a way that can be used in conjunction with the genomics and molecular data for various research projects. So that's one approach of imaging, which is retrospective, and for the data that's already collected for previous patients. But imaging can also contribute to prospectively, and PNOC and CBTTC and the various working group has done a really beautiful job in applying precision medicine treatment approaches to many brain tumor cancers. And one of the key aspects to success of these trials would be to have an imaging tool to monitor the efficacy of the treatment approaches and to modify or change the treatment if the treatment is not working, as Mike beautifully alluded to the concept of futility of treatment and having a tool to detect that and change the treatment. And in our precision medicine approach, we're going to bring together basic translational and clinical imaging approaches and integrate them into these precision clinical trials in order to predict response to treatment, disease progression, and development of resistance to treatment. And eventually, we are hoping that this will help children with brain tumor.
That was beautifully summed up. And at this point, we've made it through pretty much all of the panelists to at least, discuss at length some of the work that they're doing and how they're contributing to this. And so, I both wanted to open the end of this discussion to sort of cross-commenting on some of the information that was shared. But I would say about 5 of the 7 working groups are represented here on this panel today. And so wanted to make sure I gave an opportunity to Doctors Resnick and Prados and the rest of the folks on here who are working in some of the other working groups that we are organizing to talk maybe a little bit about some of the promising work that is happening there, and just really kind of give an overview of how else we need people to contribute, whether it's bioinformaticians or lab researchers. Again, I think we've got a really great overview of how critical patient collaboration is in this. But yeah, just kind of wanted to throw it to you two first, and then throw it back to the rest of the group for sort of closing comments.
I can maybe just briefly mention that brain tumors in children are rare diseases, thank goodness, but every child is really a critical partner in this, of course. So global outreach is another aspect of this, because when we do these clinical trials, we say we're going to enroll 40 or 50 or 60 children and try and say whether the therapy worked or not. And that may take, in a very rare disease, 2 or 3 years of accrual and analysis and decisions about whether there's been success or there hasn't been success. When we reach out to our global partners and increase the catchment of patients and have smart trials, then we can diminish the time it takes to come to a decision about whether the therapy works or doesn't work. And the other aspect of this, besides that outreach globally, which is expensive, is the protocols now. We're trying to be smarter about our protocols, and the working groups now are all working on what we call a master protocol, which becomes a living document that doesn't just start and stop, but keeps iteratively growing and learning, just like the artificial intelligence. It continues to grow and learn so that when new data comes in that reaches a threshold, we can embed it back into that protocol. So I think that that's going to increase efficiencies and the timing it takes for us to reach decisions about whether therapies work or don't work. And I just want to congratulate the working groups of thinking in a new way also about how we do what we do on the clinical trial design standpoint.
Yeah, and I can add a little bit to that in ways that, respond to your question, Jonathan. But, I'm glad you brought the global sort of lens to this, Mike, because, you can actually look at the membership of PNOC and CBTTC, and together we span more than 30 institutions that go across Europe, Asia, and Australia. We just recently launched, Haddock Australia, a platform that can support Australians' capacity to rapidly access and use data, but as well as contribute data to these efforts. And I was struck, by a recent post actually that Kristen had made, where she really was reflecting, and I'm speaking for you, Kristen, on the way that COVID-19 has been approached, in the context that we're all here today in, which is really, it's a global pandemic, right? The virus is equally infecting people if you're in China or Australia or anywhere else. It's the same exact problem for everybody. And what you've seen is this global mobilization of the exchange of data, the availability of data on behalf of implementing rapid clinical trials for patients who are facing a deadly disease. For context, right? The first viral genome from COVID-19 was made available to the rest of the world in about a week, right? That is, an unfathomable sort of standard historically in the context of the scientific process, but it's something that we have been doing, for a very long time. And so I think, one of the benefits, if there's any, in the context of COVID-19, has been sort of thinking globally about coordinating efforts on behalf of an unmet need. And that's something that, I think all of the people on the panel and all of our partners have been doing for a very long time, for which, we're immensely grateful as we move forward.
Okay.
I don't know if I characterized your post correctly, Kristen, so feel free to chime in.
Thanks, Adam. I just, basically, my post was an article of saying, globally, scientists are working together like never before, sharing data and information about COVID. And my response was, they're finally doing what we've been doing for 10 years. It was just like, this is what we've been doing. And now they catch on. And now, the attention needs to switch from COVID to brain tumors.
Right. And that's exactly, I read that same article, and my immediate thought was, if we could get that same level of input. I mean, we're already on to vaccine clinical trials for COVID-19 in what, 3-4 months? I mean, if we could spread this model of collaboration globally, what could we do for an emergency like childhood brain tumors?
And that's the key. The key has been that the prioritization has been for speed, right? And that's something so many of us constantly talk about is, speed on behalf of gaining time is what every family wants, right? So time is actually the variable that CBTTC and PNOC are trying to optimize against. That's the variable that we build infrastructure against, that we build collaborations against, that we measure success against. And I think that's really at the core of the model. Gene sort of talked about, collaborations and partnerships. Those in themselves are not novel, but when you optimize that infrastructure on behalf of time, you create new solutions, and you cannot do that on your own.
Great. And so, so many amazing comments today. I do want to just sort of do one last round of inviting our panelists to sort of make any final comments, any observations from comments of your colleagues on the call today. And so I'll just sort of open that up briefly to anyone else who would like to make a comment.
If I could tag on to something that Mike and Ali said before, is that one of the real beauties of PNOC and CBTTC has been not just discovering new therapies and bringing them to patients, but writing the protocols in a way that makes sense and making sure that our endpoints make sense. And I think Mike touched on it, that unfortunately, we can't expect every single new thing we do to work. So as long as it's not working, we owe it to the children and the families to be collecting as much information in the most transparent, efficient, and uniform way so that we can all learn from that. And when there are failures or when there are setbacks, we know how to optimize it for the next version. And we can move to a platform of making next generation studies that build off of, instead of having to scrap everything and go from scratch again. And to Ali's point, we have some new therapies like immunotherapies, which are really challenging, if not impossible, to monitor by neuroradiology, by neuroimaging, which is our standard for so many other things. So what are novel ways to get MRIs to look at new sequences to make sure that we're capturing that and we keep the right kids on the studies? Or if something is not working, to Adam's point, time is absolutely the most important thing in everything we do. Taking children off studies where for some reason isn't working and knowing that as soon as possible is critical. And so we have other partnerships with like TGen and our own genomics cores within our groups to look at circulating tumor DNA, circulating immunotherapy therapy marker DNA to see how our therapies are doing and build that into our trials so that we're really learning as much as possible. I think that we've done a great job leveraging that.
Awesome.
Thank you.
You,
So I do want to also say, and it's been alluded to a bit here. I mean, Adam mentioned in Haddock, Australia, sort of, it's a data platform that CBTTC has developed in partnership with a number of organizations. There are an incredible amount of opportunities for anyone within the sound of our voices here today to be involved. Researchers can access CBTTC data for free at any time of the year, anywhere on Earth. Patient families and foundations can hook up with some of the foundations here today, as well as some other supporting foundations of the CBTTC and PNOC that are listed on our websites. As it's been mentioned multiple times today, this research cannot happen without philanthropy. It cannot happen without regular everyday people believing in the work of these amazing professionals and helping push it forward. And so we'd like to invite you to please visit pnoc.us, please visit cbttc.org, learn how to access the data and biospecimens that are available to researchers, again, at no cost often, and to learn how to get involved with supporting or being a part of these clinical trials. And again, I want to thank each and every one of you on the panel today for being here. This was an incredible discussion. Again, we had folks listening in from across the US. As Adam said, we also have folks who collaborate with us globally. And this is such a critical message to get out. So again, thank you all so much for being here today. And thank you, our viewers, for watching.
Thanks so much, everyone.
Thanks, guys.
Thank you.
Everyone, thank you.
And
Thank you.